as much time. So we should be good. I hope you all enjoyed the icebreaker last night and the museum and the tours. Um, from what I've heard from people, they all had a good time. There were some people who were very upset that they didn't make it into the back of the panorama with Laurie. She has said that she will do it again if people are interested. So if you have some time and would be interested in going around to the museum and seeing inside the panorama, she would be happy to do it again. Um, one other note: if anybody got a parking fine for parking in the wrong parking lot, the parking lot over there, please hand it off to me and I will get it squashed. I have two already. If anybody else got one, let me know. But please park in lot 119 or 120, the one directly across here where the food truck was, or there's another one off to the left. So if you are parked in that one right now, you better go move your car because it's going to get ticketed. Okay. So with that, I will hand over to Gil, who's going to do some quick introductions, and then we'll hand over to our plenary speaker.
Thank you very much, Andy. I appreciate that. So I have been told that my job is to talk really slow so that we make this session last for our missing speaker. But I can't do that. That's not who I can do. I wanted to thank every single person for a great day yesterday. I think we had great talks, a lot of interaction, discussion. That was really super, and we appreciate that very, very much. This is the 8th year of doing the Digital Data and Biodiversity Research Conference, and we have really come to love this conference as much as anything else we do, because it really does focus on using the data that you guys all produce and sharing that data across disciplines and across domains and across a huge number of researchers. So thank you so much for being here and for making yesterday what I consider to be a super success. Also wanted to thank moderators and all the tech support people. Jill is always very good at rounding up tech support and moderators, training them, meeting with them ahead of time so they know exactly what they're going to do. And we could not make it without those folks. And I want to say that our two folks over here, Vaughn and Layla, they are fantastic. They are everywhere you need them to be at the moment you need them to be there. Don't know how they do that. And I've never quite understood, Vaughn has explained it, but I still don't get it, how he controls people in a whole nother room and he's not even in there, doing it from up here. So very good. And he also had, you don't probably know this, so he shipped 5 big crates of equipment to KU. 4 of them came to KU. 1 went to Connecticut. And so we've had some challenges getting the folks in Connecticut, the FedEx people, to get it to KU. So that didn't happen, but fortunately, he's a smart guy. He doesn't just pack one thing, he packs backups. So he had stuff in his equipment that he could still do this without any problem. I would have just packed it all up and gone back home. But Vaughn knows how to handle it. So we really appreciate Vaughn and Layla for all of their help. I think last night reception was really great. I had a really good time. It was really fun. A lot of good people, a lot of chance to talk and interact. I think that was wonderful. I wanted to thank KU Hospitality for overseeing that, making it happen, particularly Laura Hamilton, Catherine Washington, and Grace Grant. Those folks made it happen last night, and we appreciated it very much. It seemed like everybody had a good time. Some people may have had too good of a time because we have a little smaller group this morning than we did yesterday. So when they wake up, we hope they'll join in with us as well. And I also want to thank the tour guides in the museum. I know some folks didn't get back from the tours. I saw a big group go into the tours. I never saw them come back out. And then I saw all the caterers leave and all the people leave and the buses came. They still weren't back out. So I just went back to the hotel and texted everybody and said, I'll see you when you get back. And which I don't even have any idea what time that was, probably 9 or 10 o'clock. But the collections here are fantastic. And if you want to take Andy up on his offer to see some stuff today, be sure you do that. These are long time collections. Mammal collection is probably one of the first and most important in the country. And so it's a very, very good selection of collections for you to go to. Birds are also great. Also, want to thank very much Andy Bentley. Andy has been absolute stalwart in getting this set up and done. Um, he and Jill have become very good buddies, and we really appreciate that because Andy seems to know where everything is. He's always on top of stuff. You know, sometimes we work with folks and they just kind of disappear, and we don't hear from them very often. But that's not Andy. Andy stays on it and makes sure that we have everything we need. And Andy, we appreciate that very much. You've been fantastic. I also want to thank two other people, one of which will not want me to do this, but I will anyway. Um, I want to talk about Jill Goodwin. Jill is our conference manager. She is fantastic. She knows where everything is. I remember the first one of these we had way back at the University of Michigan, and whenever it was, 8 years ago. And our director then said, "She's pretty good, isn't she?" I said, "Yeah." And that was the very first one. So, I mean, I think it's very important. I don't think it's, for me even, as well as I know Jill, as much as I work with her, I still have no real idea what she does and how she gets all the stuff accomplished that she gets accomplished in planning these conferences. I'm kind of slack. And when we have a workshop, we have a few meetings and we do something. Jill, she meets with everybody ahead of time. She calls in all the presenters. She makes sure that everybody has what they need. And that is absolutely fantastic. So she is our conference manager and has been super. And Jill, thank you so much for and working with her. I don't know where Kit is, but she's here somewhere. Kit Lewis, Kit, are you in the room? She's out working. Um, Kit Lewis is Jill's assistant, and they have put a lot of this on together. And Jill and Kit has done a whole bunch of the stuff for SCED, getting all the schedules up, making sure that everybody's in there and their names are spelled right, and all that sort of thing. I did notice that this session was supposed to start at 10:29, and I'm not really understand. I was looking on my phone, and the schedule said 10:29. I thought supposed to be 10:30 or something. Or the one that I'm going to next. And so anyway, but if that's all we did wrong, that's not too bad. Okay, all right. Yeah, never mind. No more critical comments. Um, I also want to very much thank Jorge Soberon, who is the current director of the Biodiversity Institute here and has been very, very complimentary in having us here and wanting us to be a part of this. Um, Jorge is on his way to retiring from being the director, which I'm going to personally miss very much. But he's being replaced, as he said yesterday, by Nico Franz. And he, Nico, he's not yet, he doesn't until July 1st. So we can't say to him, he is the director. However, Nico was the director at ASU last year when we did this conference at Arizona State University. So Nico, this is his second year, and plus he's very tied into IDIC Bio as a co-PI from the Symbiota Support Hub, and we really appreciate Nico's a broad thinker and a quick thinker, and we appreciate all the work that he has done to help us pull both of these off, and so we want to thank him too. Okay, so we're ready, I guess, to start. Thank you so much, folks, for being here. We'll have a good day today.
Alrighty, so let's get this party started. Our plenary speaker this morning, oopsie daisy, I just moved all of that. Put it back where it was. There we go. Our first plenary speaker this morning is Joanna Slusky. She's a professor in molecular biosciences here at KU, and she's going to be talking about outer membrane proteins and AI. Joanna, all yours.
So I noticed that the plenary speaker that we're going to be missing today is connecting to other disciplines, and that will very much be a theme today because I am a computational biologist, a biophysical chemist, and so this might be somewhat different from what you've been hearing about so far, and so I thank you all for welcoming me to be part of this community today, and thanks especially to the organizers for inviting me to give this talk. So as Andy said, I'm Joanna Slusky. I'm a professor here, molecular biosciences and computational biology. And so as one of the people here from Kansas, I'd like to welcome you all here as well. And if you'd like to talk about where to get great cocktails or where the local bridge club is or things like that, I'm happy to discuss it with you. Now, I think about proteins, which is definitely on the micro scale compared to a lot of what you've been hearing about so far. But I want to tell you about why I get so excited about proteins and then about the specific proteins I get excited about. The specific proteins I get excited about, just as a quick mention, are these barrel proteins. They're sort of tube shaped. They're in the membrane, and in my lab we think of basically playing cat's cradle with them, which is what I'm showing here. So I'm gonna make this small. See me? I don't need to see me. Okay. So just to sort of step really far back, there wasn't so much talk about proteins yesterday necessarily, but there was talk about genomes. And just so you remember, when we think about all the information that we have to encode about a biological organism, that's going to be in the genome. The DNA fundamentally encodes for what amino acid sequence gets made, right? That's translation, right? The DNA goes to RNA, the RNA gets onto a ribosome, the ribosome makes these chains of amino acids, and these amino acids ultimately become proteins. So you've been thinking maybe about the DNA as encoding all of this information, and that's true, but it's layers and layers of information, because fundamentally, when I look at this amino acid sequence, some amount of, one of 20 possibilities that are in a chain linked one to the other sequentially, right? That's what you're getting out of this DNA. And that fundamentally encodes a protein structure. So each sequence of amino acids generally only creates one structure, and that one structure could be all kinds of different things, but these are the proteins that will then fundamentally do all of the chemistry in all of the cells. And the structure is what tells you what kind of chemistry, what kind of function you can accomplish. So if you have a hand-like protein, you're like, oh, that's a protein that grabs things. Or you have a door with a hinge and you say, oh, that's what allows things to get in and out or stops things to get in and out. As a protein scientist, I look at protein structure and I say, oh, that tells me what it does. But how do I get there? Well, that's a question for what exactly is that sequence, and getting from sequence to structure is somewhat unknown, but then ultimately knowing the structure tells us a lot about the function, right? And so that's how I think of these levels of information. And so I'm super excited about different types of proteins because this will tell me what they do and will give us some sense of what a cell can do with those proteins. Okay. So a lot of people like to say it's not what's on the outside that counts, it's what's on the inside that counts. We go in the other direction in my lab. We say it's what's on the outside that counts. And that's not because we're particularly interested in our physical environment. In fact, I get the impression from many of you that you really like to touch the physical environment much more than I do. But really, we look at the outside of bacterial cells and we say this is really where it's at. If I'm going to design a new protein, I want to do it here. So this is a watercolor image from David Goodsell of a cutaway of a bacterial cell. Bacterial cells will often have two membranes. So this is the inner membrane over here, and then this over here is the outer membrane. Okay, and that outer membrane is what has these proteins that I'm interested in. So these would be the proteins. It turns out, unlike many other membranes, the outer membrane of bacterial cells, almost 50% protein by mass, quite a lot. And so these proteins will control the interaction between the cell and its environment, and that makes it a very powerful chemistry position because it allows us to deal with all kinds of problems that we have, such as antibiotic resistance. So, right, the structure determines the function. There are all kinds of functions for these proteins, and this kind of protein over here, that one pushes antibiotics out of bacteria. That's a big problem for antibiotic resistance. So if we could plug that, we could stop antibiotic resistance. We would also be interested in making new enzymes at the surface of cells. That would allow us to potentially degrade pollutants without having to bring the substrate in, without having to push the substrate out, just doing the chemistry at the cell surface. We're also interested in membranes because we could use them for various types of biosensors. You could have a protein across a membrane, and as I'll show you, that could be various radii that could coordinate different metals. You could then get a voltage across the membrane that would change depending on whether or not it binds a metal, and so you could have a heavy metal biosensor using that. So we have all of these proteins at the cell surface. We can make new ones, and that's very exciting to me and my whole laboratory. Now, we get excited about this position partly because of the position and all of the things it can do, but partly because of the shape. So I told you at the cell surface, we're going to need to do all the chemistry that a cell needs to do to interact with its environment. But all of these proteins, as it happens, and this is super unusual in biology, they all have the same shape. So if you're not used to looking at proteins, I will try to sort of show you a little bit about what we're looking at. But this is an outer membrane protein that causes cells to stick to other things. It calls an adhesin. This is an import protein. A lot of membrane proteins are used for import and export. This is sort of the hallmark of membrane. So this would be an import protein. You're going to import nutrients. This would be an export protein. This is the one that exports those antibiotics that we're so concerned about. You also have membrane proteins that cause other membrane proteins to ultimately reach their fold and get into the membrane. That would be an assembly protein. I'm going to talk more about that later. Bam. And here we have an enzyme. And what you're looking at, if you look between the blue lines and the red lines, those aren't part of the protein. That's the boundaries of the membrane. That's how we denote the boundaries of the membrane. And so if you look between those, what you see is that unlike some of those other pictures of protein structure that I was showing earlier, these are all beta barrels. So there's no alpha helices in that region. Those would be the two different options for protein structure, alpha helices or beta sheets. But it's completely beta sheet, and they wrap around to make this barrel tube-like structure, right? So they're all up-down beta barrels. So the beta strands all go up and then down and then up and then down. And they're all right-handed. You can see that they're all sort of tilted in the same way with respect to those boundaries of the membrane. And the only difference really is that you've got a difference in strand number. So that's how many of those arrows that are crossing the membrane, and those would have a linear relationship to the overall diameter of that pore, right? Because circumference and diameter have a linear relationship, and that's what's creating the circumference. So you've got this change in strand number. If you could change the diameter, again, that would allow you to coordinate different heavy metals for possible biosensors. It's also the case that barrel structures make great enzymes. Enzymes are those proteins that do chemistry. One of the things that you'll need for an enzyme is to be able to sequester substrate from solvent, and barrels make a hole, which does it really well. So it's a great fold and a great location, and there's a lot that nature does with that, and there's a lot that as a protein designer, my lab does and thinks about doing with this kind of protein. So When we think about collections, I think about collections of these proteins. And so my lab started out with what was the collection of these proteins and what could we say based on that collection and about the evolution of these proteins based on that collection. And then we substantially increased this collection by developing an algorithm that used homology data, as I'll explain to you. And then we were able to go from that to be able to generate a tremendous collection, 2 million proteins, and now we're using that collection to do generative AI design of new proteins. So how did nature evolve this one structure to have so much function, right? I said that this structure can do everything that the cell needs to interact with its environment. Lots of function, one structure. And so to some extent, we use the paradigm of convergent versus divergent evolution to think about this. This may be more in some of your wheelhouses, but just to make sure we're on the same page with respect to this paradigm, you can have similar sequences for proteins and you can have similar large-scale macro structures, and those will all point to divergent evolution, evolution from a common ancestor, right? So human hands and cat paws and whale fins and bat wings might all have similarly articulated ulna and radius bones, whereas you could have convergent evolution where you have a similar structure, but ultimately quite different because of maybe differently articulated ulna and radius bones and different sequence homologies that will ultimately indicate that there was sort of multiple events that got evolution to the same place, and that would be convergent evolution. So these beta barrels, these outer membrane proteins, are often thought of repeat proteins. They're known that there are, I talked about the strands, those are the numbers of beta strands per barrel, but about between 8 and 36 known. And so if the motif, the structural motif is then this beta strand, a loop, and then another beta strand, you could imagine that this is genetically getting copied and pasted some number of times to create a whole barrel. Okay, and so that's often the paradigm that we think of. And so the question then where we started was, is this kind of duplication event how we get the whole diversity of this fold? So we started by making something of a network model. It's a little bit complex, but what you're looking at is all the crystallographically, structurally resolved membrane barrels. So just a little bit of background, how do you get protein structure? X-ray crystallography. More and more, it's happening through computational methods, and I'm happy to discuss that with you probably separately. But we have structures. These are ways of getting crystals of proteins and then figuring out exactly where all the atoms are. And so we know both now the structure of some proteins and the sequence, the amino acid sequence of those proteins. And we can look at those, and each of those in this picture would be a colored rectangle. So you've got these colored rectangles. Those are all the structurally resolved beta barrel proteins, and there exists an edge between each of these colored rectangles, between each of these nodes, if there is a certain amount of sequence similarity between them. And we're judging the sequence similarity by an e-value. This e-value would tell us if there's how many false positives for every true positive. Here, we're looking at about 1 false positive for every true positive. So we're trying to cast an extremely wide net so we can have the sort of broadest definition of what could be related. Later, as we're trying to exclude possibilities of relationship, we might look at lower e-values. 10 to the minus 5 would be 1 false positive for every 10 to the 5 true positive. That's going to be a lot more reliable, things like that. So what you'll notice is that there seems to be a couple of different unrelated clusters. These are these smaller clusters on the outside, so possibly indicative of convergent evolution onto this fold, but that most of the beta barrels are related to each other. This is what we call the prototypical barrels. And that's likely indicative of some divergent evolution, that these are all from a common ancestor. And so we wanted to look at these more specifically to understand this question of, were these duplication events really the majority way of the diversification? And so the paradigm here would be looking at duplication events versus mutation events, where we could say, in biology, especially in the biology of bacteria, duplication events are much more common than these kind of mutation events. And so we would expect to see basically variations in the number of strands, right? That's what I told you are getting duplicated, these strand loop strands. And mutations would mean sampling among a specific strand number. So if we recolor this green grouping by strand number, so we have the structures, so we know exactly how many strands each of these proteins has, we can say, okay, here's the 8-stranded barrels, 10, 12, 14, 16, 18, and 22 stranded barrels. And what you see, and we've got a GIF going where it's going between e-values, so you can see more and more closely related proteins, that things are more and more closely related to the same strand number, and you're not seeing a lot of bouncing around among strand numbers. So what that's going to tell you, right, is that you do have a lot of mutation events, more than you would expect, and that biology is somehow making fewer possible duplication events. At the same time, you do see internal repeats, alignments between proteins that you can get within the protein, which then shows that, in fact, you do have these duplication events. And so you can see the number of proteins with internal repeats on the y-axis, and of the different strands per barrel. Lots and lots of them have these internal repeats, these self-alignments, which would then indicate that on the one hand, there are these infrequent duplication strand accretion events which control strand number diversification, but overall the mutations seem to dominate the diversification of these prototypical outer membrane beta barrels. So we went from here saying, okay, this is what was known because people had structurally solved about 200 outer membrane proteins, and we said, okay, well, that's for 200 of them. What can we know if we see more? So with more barrel sequences, could we know more about evolutionary mechanisms of this particular protein fold? And could we know how this protein fold actually achieves its structure in some way? How does it get into the membrane, things like that? So to do this, to make a larger data set, we used a trick using sequence coevolution. So the way this works, and now I'm depicting homologs, which you would usually see as a string of letters, as widgets, okay? And strings of widgets. And so we've got 4 homologs here. Usually we're going to use thousands of them. And we can say, okay, there are positions among these sequences that are covariation, that are mutating at the same time. So if you see a position like the first position and last position where they are covarying, that often means that they are near each other in physical space. So that allows us to go from a set of sequences to physical representations of where those atoms actually are. And so that concept will ultimately create what we call a contact map, or an evolutionary contact map. This is not a real contact map. We don't actually know where the atoms necessarily are, but residue is just another word for amino acid. You have the amino acids of the same protein both on the X and Y axis. So you've got the self along the diagonal. But then what you're looking at is, here would be the first set of amino acids interacting with the second strand of amino acids. And you'll see these, and this looks quite different for a contact map of a bunch of alpha helices or alpha helices with beta sheets. A beta barrel looks like this and only like this. So we've got one strand interacting with strand 2 interacting with strand 3 interacting with strand 4. That's what these lines along the diagonal ultimately mean. And you keep going down until you get to the corners where the first strand interacts with the last strand. So that's right, this strand interacting with that strand. So you have these very distinct evolutionary contact maps, and from that, we were able to create a fairly simplistic algorithm to identify one type of structure from all different types of sequences. So that algorithm was developed by, at the time, a high school student, Rebecca Bernstein, in my lab, where the algorithm just said, is there this closing contact? That's what we call when the first strand and the last strand interact. If so, it's a transmembrane beta barrel. If not, we can count the number of these other strand-strand interactions, these hairpins, and if you have enough of them, at least 4, it's probably also a beta barrel. And this fairly simplistic algorithm got us a 96% balanced accuracy, extremely low false positive rate. We could compare it to other models, and for those of you who develop machine learning models or other algorithms, we're using an MCC, that's a Matthews correlation coefficient. What we have here, because we have so few structurally solved outer membrane proteins compared to large numbers of non-outer membrane proteins, it's a very imbalanced data set. And so when you're doing any kind of training or doing any kind of metrics about how well you're doing, if you just use accuracy, right, if you never predict an outer membrane protein, you will still get like 90% accuracy because of how accuracy is calculated. So with a Matthews correlation coefficient, you can use the true positives and the true negatives and the false positives and false negatives to get this overall truthiness score of what are you predicting, and we get a MCC of 0.921, which is very high, and I'm showing the true positives and true negatives here. We can compare it to any other sequence method of figuring out if it's a beta barrel, BOMP, the Freeman-Wimley score, PRED-TMBB. We're doing better than all of them, and fundamentally, I'd just like you to notice, we're doing specifically much fewer false positives, which means that when we create this data set, we can really believe that the proteins that we say are barrels. So we have a really high confidence level in that. Okay, so then we can answer questions like, wow, how many outer membrane proteins per organism? So if you go to the NCBI database and you're able to get all kinds of genomes of all kinds of bacteria, we can just look through those bacteria and say, now we know how many outer membrane proteins that bacteria has, which was previously unknown or certainly undercounted. So we started with about 3,000 reference proteomes, and this was work that was done by a postdoc in my lab, Daniel Montesano, where we take barrels from previous data sets. So people had previously annotated some of the barrels, and we actually figured out that people were way overcounting barrels because they were using these other methods which generally had tons of false positives. And then we could identify the phyla or class that they belong to. And so we've got numbers of transmembrane beta barrels on the y-axis, and the distribution when looking at 3,000 proteomes for all of these classes or phyla, Bacteroidetes to Firmicutes, and we can see that people were generally counting about 70 or fewer transmembrane beta barrels as the varieties of these outer membrane proteins. And then we could search the proteomes for more using this is it a barrel algorithm. And what we found was substantially more barrels. Things that were unrelated to previously known barrels. So you can see fold increase on this other y-axis over here. It's in log. So you can see that right now Bacteroidetes have the most. We've got, sometimes up to 200 or more types of transmembrane beta barrels per organism. Firmicutes have the least, but we're getting 50 times more than previously predicted. So we're seeing lots and lots of different types of barrels. So then we could expand the data set beyond those 3,000 reference proteomes because we could take sort of 30,000 sequences, some of which were barrels and some of which were not barrels, and we made all the pairwise comparisons between those 30,000. And those we had already looked at the contact maps and already established whether or not they're beta barrels. And so now what you're looking at on the y-axis is the same is it a barrel prediction? Are these both barrels? Are these both not barrels? Or is one a barrel and one not a barrel? And you can compare that to sequence similarity. And what you see is that there's not a close relationship from 0 to 40 to 0 to 30, 25% sequence similarity, totally unrelated if they're both barrels or both not barrels. But as soon as you hit the 40% mark or so of sequence similarity, either they're both barrels or they're both not barrels. So now we can have an easy metric of saying, okay, at 40% sequence similarity, sequences have about 98% likelihood of being either both barrels or both not. So let's take a bunch of sequences. Those are the purple dots here. And let's cluster them, see how related they are. Those are fairly well-established algorithms. And then if we have these representatives, those are the slightly bigger ones, if we predict all the representatives, if we generate all of their contact maps and then put them through our algorithm, then we can predict all of the things that they're related to and assign them. And so when we do that, we get about 1.9 million outer membrane protein sequences ultimately. This is the largest data set of outer membrane protein sequences, and we have extremely high confidence in it, as I told you, because of this low false positive rate, so we can do a lot with the information. So the first thing that I'll mention is that we can then say, okay, among these groups, I showed you this picture before, just based on sort of the 200 structurally solved barrels, where some seem to be unrelated. Now we can look at these 1.9 million and see what's related. And when we group them into related or unrelated types, we find that there are 11 separate types. These seem to mean that there have been at least 11 instances of convergent evolution onto this one fold that is always in the outer membrane, this beta barrel. 6 of these types had never been structurally resolved at all, and 3 of them had never even been annotated within UniProt, which is this large database of proteins. So we can look at things like domain of unknown function 1302. This we now know are these outer membrane beta barrels, but we don't yet know what they do, or hypothetical protein type 1 or hypothetical protein type 2, etc. So from this information, from these 11 types, we can identify what the barrel size is, and we can do that again from the contact map because we know where that closing contact is. And if you know where the closing contact is, that means you know where the first strand and the last strand is, which means that you can estimate the barrel size of amino acids. So we've got estimated size versus the actual size that's been resolved in a crystal structure, and you can see that the R squared is fairly good. So then you look at the 11 different types of proteins, and you say, what sizes are the barrels? And number one, I guess you can say, okay, some of them have not evolved very far. You had one type of barrel, it exists as a barrel, and it doesn't evolve any further. But then you could look at these other types and actually see evolutionary duplication events just in the amino acid size of the barrel. So for example, if you look at the prototypical barrels, you can say, well, 161 is about half of 333. That looks like an evolutionary duplication event where that whole barrel was just copied and pasted to make a larger barrel. But you can also see that 242 is exactly half of 484, or 232 is exactly half of 464. For these larger groups of barrels, there seem to be very, very clear duplication events, like we were anticipating. The other thing, and this is going to be a little bit more abstract to you possibly, because you might not be used to thinking about sequence motifs and amino acid sequence motifs and what those do chemically. But I told you before that there's this assembly beta barrel that causes all the other beta barrels to be able to get into the membrane, or so we believe. And that one is called, excitingly, BAM. Okay, it stands for beta assembly machinery. And what you're looking at here is work by another group, the Bernstein group, where you've got a outer membrane protein that's the client protein that's being inserted, that's in red, and the guy that's doing the inserting, the barrel that's doing the inserting, is in blue. So on the red protein, there is this green strand, which is called the beta signal, and people know that it has a particular sequence that is able to bind to the insertion protein in blue. And the sequence goes like this. Even for me, this is an unusually hard thing to wrap my head around, where it's got some number of polar amino acids and glycine and aromatic amino acids and hydrophobic amino acids with some undefined amino acids. So we said, well, can we find, now that we have this 1.9 million protein database, what do we see with respect to these kinds of sequences at the end of the protein that might get used for insertion and interaction with this insertion protein? So what we see is that all of the different 11 groups have sequence motifs. So what we're looking at here, I don't know if you're used to seeing logos plots. This is generated by a algorithm called MEME. The logos plots have bits of information on the y-axis, and the larger and higher up the different letters are, each of the letters stands for one of the 20 amino acids. The larger and higher up the different letters are, the more of that you have at a particular position. And these kinds of algorithms are able to align lots of different proteins to find a particular motif, even if it's not all at exactly the last amino acid. It finds where things are similar and can give you these motifs. And so what you see is that they all have motifs, but only the prototypical actually has what's considered that classic beta signal that I was telling you about before. And it turns out that that classic beta signal is actually much, instead of just looking at it as sort of, polar, glycine, aromatic, what you see is that actually it's quite clearly sort of an LGL GYRF motif. But that there are these large differences with these other ones where they don't have a classic beta signal, but do still have a signal. And so then the question becomes, do these get inserted with that BAM complex, or do they use some other complex? Do they have some other method of using the BAM complex, or do they self-insert? And that's something that we don't know, but we're actively interested in understanding, how do these proteins get into the membrane? What is their story of biogenesis? And so I'll just sum up from here. We have this data set of 1.9 million bacterial outer membrane proteins. This is the largest data set, most accurate way of identifying outer membrane proteins, and we're updating it with more information all the time. But we keep this at isitabarrel.ku.edu, and you're welcome to search through it. We have a number of search mechanisms for it. So about 3.5% or less of bacterial proteomes are these outer membrane proteins, which all have the same barrel shape. We see that clustering of protein lengths is, again, suggestive of this sort of diversification events of evolutionary duplication. That we find 11 different types of these outer membrane proteins, 3 of which had not even been previously annotated, and that the diversity of signal motifs by type indicates a variety of insertion mechanisms that we still don't yet know about. And so just to sort of tell you where we're going with this, because I figured if I was going to tell you about the set, I wasn't going to have time to tell you about the machine learning that we're using to generate. But we have this truckload of beta barrels, or like this Twitter user said, truckload of beta sheets, and 1.9 million proteins with the same fold. The sequences are extraordinarily different, and that's partly because multiple classes are consistent with convergent evolution. Told you about the 11 classes. Also, in general, bacteria just diversify like crazy, in part because they've got extremely short generation times, in part because they have extremely high mutation rates because of terrible proofreading. And yeah, membrane proteins tend to mutate more than other proteins. I didn't tell you about this, but things at the surface of the cell tend to mutate more, which means that our proteins have super diversified. And so we have extreme diversification within protein sequence space on one particular fold. And so we've been using that to train various types of machine learning algorithms for generative design, partly using LSTMs, also GANs, also transformers, and generative infilling, again using transformer architectures, fine-tuning ESM architectures for classification. So that's another transformer task. But all of that I'd be happy to talk with you more about. And I'd like to thank the people who did this work. I spoke about what Daniel Montesano did. I spoke about what Ryan and Megan did at the beginning with the network model, Rebecca Bernstein's algorithm. This is the beginning part was in collaboration with Margarita Saji and Rachel Kolodny at the University of Haifa. And I'll just point out that Emily Proctor in my lab has been working on doing this for chloroplast proteins, which also have an outer membrane. And we're very interested in that, but I know a lot less about plants than I do about bacteria. So if anybody wants to talk with me about chloroplast outer membrane proteins, I'd be extremely excited to do that. I'd like to thank my funders, and I'd like to thank you all for listening.
Thank you very much, Joanna. Any questions for Joanna? Or you're all stunned into silence?
Thank you. So my question is, could you kind of go into a little bit more detail about how you go from a sequence to one of those contact maps?
Yeah. We use a program. There are a couple of different programs that do this. Gremlin does it. AlphaFold has a version of it, I would say. We use Raptor X. We find that all of these different contact map programs and the coevolutionary algorithms are designed to work on all proteins. But I think some algorithms work better on some folds and some work better on other folds, even though they're all trying to be for all proteins. And Raptor X for us, which is developed from Jinan Bao's lab at Chicago, that's what we use and that's what works best. Yeah, thanks.
I want to say thank you. That's very exciting. And I love all the graphical images as well. I think they have a great potential for exciting a lot of us to ask a lot more questions. It's been a long time since I took classes related to this, but I remember alpha helices and beta sheets. My question is, can you give us a couple of concrete examples of applications? So I started thinking, if we understand something more about the way in which transport's happening, is this avenues for pathogen control? Is this ideas for stopping things from getting in or making things come out? Or is that the doorway that we're trying to block or insert?
Yes. So the applications that we think about most in the lab are if we understand how things fold into the membrane or don't fold into the membrane, or what's specific for good bacteria, bacteria that's good for us, or bacteria or pathogenic bacteria, then could we develop new antibiotics? Could we develop antibiotic potentiation mechanisms that existing antibiotics could overcome existing ways of methods of resistance that the bacteria have evolved. So that's sort of from the kill them all. But then we also think, right, about like, could we make, I told you that this type of protein would be great for enzymes. It already makes lots of enzymes, but could we make our own types of enzymes at the cell surface so that we could degrade more pollutants, degrade oil spills, things like that? Could we make these types of proteins and again, vary their radii based on their strand number and have particular coordination geometries for the metals so that we could have biosensors that would ultimately be able to bind different types of heavy metals? You could be able to tell what types of heavy metals are in your water, for example, and be able to do that with a simple voltage output across the membrane. So those are what I think of as the applications, and I think being able to understand how they fold, being able to understand how we can make new ones, being able to understand how these are both sort of scaffolds but also active, chemically active parts of proteins, and be able to move between those, I think would be very powerful.
Marvelous. Thank you.
Thanks. That was crazy and fun. I'm not a geneticist, so I have a database question.
Yeah.
So I was playing with your website.
Oh, thanks.
And
did it break yet?
No, it hasn't broken yet. There's a button at the bottom that says download database. And it says it's 1.4 gigabytes. Do you know anything about how the database is held, what it's held in, how would I keep track of versions? Like if I grab it today and then you put more stuff in it tomorrow.
Yeah, we're not so good with versioning right now.
No, it's not a criticism at all. It's just like because I was playing.
I think it's a CSV file. Is that what we're?
It looks like it.
Yeah. Okay. Good. I think that we're sort of putting it into a CSV file. We have not been careful about versioning. I think as we're just moving into, we just developed an algorithm to identify the number of actual strands each barrel has, which is not trivial. I showed you the very beautiful contact map. A lot of the contact maps are less beautiful. It's less, we said we'll take anything that has 4 strand-strand interactions, which really means that it probably has a lot more because you can't always see the strand-strand interactions. So how do we ultimately determine more structural information? So we have an algorithm now that can incredibly well predict how many strands each of these, or at least 500,000 of the 2 million of them, have. And so then we wanted to put in a separate, we're going to roll out a separate version. We haven't thought about then, well, will we have the original version available also? I think that's a great question, and probably we should, but we haven't quite dealt with that yet. And it's a good thing. I appreciate you bringing that to the forefront of my mind. Do we have a question over the corner?
Thank you. Thanks for your talk. I really enjoyed that. So I have a little bit of a comment and then a question, and I may have misunderstood something, and so then the rest of everything downstream from that won't make sense. So you can just correct me if I'm wrong on that. But one of the things I think I was really struck by was your emphasis on the role of duplications versus substitutions. And I think that's something we see at the genome level as well.
Absolutely.
And in eukaryotes as well as in bacteria. And so it seems that a lot of innovations in eukaryotes actually arise from having duplicated genes and duplicated genomes. And the more I've been reading about bacteria, I've been finding that there's a lot more polyploidy in bacteria than people suspected before. So I'm wondering about the sources of the duplications, if they come from duplicated genomes or if they come from duplicated genes within a single genome, or what the source of those duplicates might be.
Yeah, it's a good question, and it's not, as a chemist, it's something that I sort of read about and speculate about, but know a lot less about those bacterial mechanisms and genetic mechanisms. So what I'll say though is, if what we're seeing is not whole genomes, and frankly not even whole genes being replicated, but pieces of genes ultimately being replicated, and that we see a lot in different forms of bacterial proteins, then it must, Andre Lupesh calls it strand accretion mechanisms, or domain accretion. You see this a lot, where domains will get moved from one protein to another. Suddenly, a protein that only has one domain existed in another organism, and it has 2 domains, which were previously 2 different proteins or something like that. So I think it's a lot of these small, what we're seeing here, certainly for the individual protein evolution, we're seeing some sort of sub-level of duplication. Now I'll just, since the other speaker ceded all of her time to me, I'll just note that if you look at these 11 different types of proteins, and we try to sort of map out these phyla, and maybe some of you have strong opinions about this, by how related they are to each other in order-ish. And so you can see, and this is based on a lot of work by Simonetta Gribalto, but but the idea of these is sort of in order, and it's almost checkerboardy. Look, all of them have prototypical proteins. This is just the number of the type of protein that they have. All of these phyla have LPTD-like proteins. After that, it becomes really a checkerboard, so that the more related the organisms are, in no way determines whether or not their sister organisms, their sister phyla, have the same types of these outer membrane proteins. We suspect that there's a ton of horizontal gene transfer that these guys are involved in. And so what we're dealing with evolutionarily is both these accretion mechanisms of bigger and bigger barrels where just copying and pasting small genetic elements, and then horizontal gene transfer. Yeah.
Any other questions? Don't see anything online, so I think we're good. Thank you so much, Joanna. There was some great food for thought.